Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine this. Your body has a super smart way of knowing if something's not right in your gut, even if it doesn't directly see the troublemaker. Think of it like maybe a home security system that detects an intruder by the weird sounds next door.
That's a great analogy.
Well, today we're diving deep into exactly this kind of clever detection system happening inside you.
We are. We're looking at a really fascinating scientific study. It's titled Cross-Kingdom Mediated Detection of Intestinal Protozoa through NLR Family Pyrin Domain Containing 6.
Okay, that's a mouthful.
It is. But basically, this research, it blends health news insights with some really cutting-edge science. And it shows us a, well, frankly surprising way your body spots certain invaders in your intestines.
And the research points to this incredible interplay, right, between all sorts of tiny organisms living together down there. You got protozoa, these single-celled things, then bacteria, and of course our own immune system.
Exactly. And right in the middle of it all is this key sensor. Scientists call it NLR Family Pyrin Domain Containing 6.
NLR Family Pyrin Domain Containing 6.
Got it. And it seems to play this vital role in keeping things balanced, but, and this is key, without directly attacking the protozoa themselves.
So, okay, our mission in this deep dive then is to really unpack how this sensor, NLR Family Pyrin Domain Containing 6, can indirectly sense these protozoa.
Yeah, and what happens when it does sense them? What are the consequences? And, ultimately, what does this mean for how we should think about our gut health?
All right, let's get into the study itself. The researchers, they were tackling a pretty fundamental question, weren't they?
They really were. Essentially, how does our first line of defense, the innate immune system, actually identify when there are protozoa, these specific single-celled organisms, in the intestine?
Because the way we thought it worked wasn't the full picture.
Exactly. The mechanisms weren't well understood at all.
But what did they find? What was the observation?
Well, something really interesting they saw was in mice infected with a type of protozoa called Tritrichomonas.
Okay. Tritrichomonas.
These mice, they developed a noticeably thicker layer of mucus in their colon.
A thicker mucus layer. Interesting.
And crucially, this thickening depended specifically on that sensor we keep mentioning, NLR Family Pyrin Domain Containing 6.
Ah, okay, so it's directly linked.
Directly linked, and also on a couple of other molecules called apoptosis-associated speck-like protein containing a card and caspase 11.
Right.
And all this pointed towards the activation of these specialized cells that line the gut, called goblet cells. They're the ones that make mucus.
Goblet cells. Okay, but here's the twist, right? Because it wasn't the protozoa itself causing this.
Precisely. This is where it gets really clever. The mucus increase, it wasn't triggered if they just put purified Tritrichomonas protozoa into the system on their own.
Huh. So what did trigger it?
It only happened when they used extracts from the cecum, that's a specific part of the gut, from mice that were already infected.
Wow. Okay, so that really suggests that NLR Family Pyrin Domain Containing 6 isn't, like, directly seeing the Tritrichomonas?
Exactly. It strongly implies it's sensing some change in the gut environment caused by the infection rather than the bug itself.
So, connecting this to the bigger picture, what were those changes? Did they figure that out?
They did. They looked deeper into what these infection-induced changes might be. And they found that when Tritrichomonas was present, there was a definite shift in the gut microbiota.
The whole community of microbes living there.
That's right. Specifically, they saw an increase in certain types of bacteria, particularly ones called Bacteroides and Prevotella.
The Bacteroides and Prevotella. Okay, so the protozoa infection leads to more of these bacteria.
Seems like it. And the story gets another layer deeper. They did what's called a metabolomic analysis.
Looking at all the small molecules.
Exactly, all the metabolites present. And they found a significant jump in various molecules during the infection, including a type of fatty substance called sphingolipids.
Sphingolipids. Okay, so we've got the protozoa, Tritrichomonas, leading to more Bacteroides and Prevotella bacteria, which then leads to more sphingolipids.
That's the picture emerging. So naturally, the next question is, how does this all connect? How does NLR Family Pyrin Domain Containing 6 fit in?
Right. What's the mechanism?
So they used some really neat approaches here. They used germ-free mice with no gut microbes at all.
Okay, a clean slate.
Precisely. And then they introduced specific bacteria, and what they discovered was pretty striking. a particular bacterium, Bacteroides the Tioteomicron.
One specific type of bacteroids.
Yes, Bacteroides the Tioteomicron. Just that one type is actually sufficient to trigger that NLR family Pyrin domain containing 6 dependent response in the goblet cells.
Wow.
But, and this is crucial, only when Tritrichomonas was also present.
Ah, okay. So
Yeah.
you need both, the protozoa and this specific bacterium.
Seems so. And even more telling, when they used a modified version of that same bacterium, one that couldn't produce sphingolipids.
Let me guess, the effect disappeared.
You got it. The effect didn't happen, which strongly implicates those sphingolipids.
Fascinating.
And just as a sort of side note, they also observed this whole process seemed to be more pronounced in female mice compared to males.
Oh, interesting. We'll have to come back to that.
Definitely something that needs more looking into.
So, okay, let's piece this all together. The researchers are basically proposing a completely new idea, aren't they?
Absolutely. A really novel concept. They're suggesting that NLR family Pyrin domain containing 6 is acting as an indirect sensor.
Indirect. So instead of seeing the protozoa,
It's monitoring the levels of these microbial sphingolipids, which increase during the infection, probably because those Bacteroides bacteria are multiplying.
so the bacteria are like the messengers, signaling the presence of the protozoa through these fatty molecules.
That's the proposed model. It's a remarkable example of, well, cross-kingdom communication happening right there in your gut.
Cross-kingdom, meaning between different types of life, like protozoa, bacteria, and us, the host.
Exactly. You have a sensor from our body, NLR family Pyrin domain, containing 6, detecting one type of microbe, the protozoa, by sensing chemical changes produced by another type of microbe, the bacteria.
It's like our body's eavesdropping on the microbial chatter.
You could definitely put it that way. And it's not just listening, it's acting on that information.
Right, because this NLR family Pyrin domain containing 6 response, it leads to more mucus. Why is that important?
Well, that mucus secretion seems really crucial for maintaining a strong barrier. When these protozoa are around, that thicker mucus layer could act as a kind of physical shield, keeping potentially harmful organisms away from the gut lining.
Strengthening the defenses. Makes sense.
It does. And the whole chain of events, they uncovered, the Tritrichomonas infection, then the increase in Bacteroids, the rise in Sphingolipids, and finally the NLR family Pyrin domain containing 6 response. It provides a really elegant potential explanation for how our immune system can handle the sheer diversity of threats in the gut. It doesn't necessarily need a unique receptor for every single bug out there.
It can just monitor the overall neighborhood, the microbial environment, for signs of trouble.
Exactly, by monitoring these metabolic byproducts like sphingolipids.
So what does this mean for, us, for our understanding of gut health day to day?
Well, I think the finding that these sphingolipids produced by really common gut bacteria can trigger such a specific protective response, it opens up a whole new perspective on gut balance or homeostasis.
Not just about good versus bad bacteria then.
It seems much more nuanced than that. It's about the complex interactions, the signals they send, the metabolites they produce. It's a dynamic ecosystem.
And does this have implications beyond just protozoa infections? You mentioned cancer earlier.
Yes, that's another significant part of this. The study looked at a mouse model for colorectal cancer, and they found something quite concerning.
What was that?
In mice that lacked the NLR family Pyrin domain containing 6 sensor, if they were also colonized with the Tritrichomonas, their tumor development actually got worse.
Wow. So lacking that sensor made things worse in the presence of the protozoa.
Correct. But importantly, this didn't happen in similar sensor-deficient mice that didn't have the protozoa.
Okay, so the protozoa plus the lack of the sensor was the bad combination.
It appears so in this model, which really underscores how important this indirect detection mechanism might be, not just for fighting infections, but maybe even in preventing or mitigating other serious diseases like cancer.
Yeah, that's pretty significant. But like all good science, we need to talk about the limitations too, right? This study isn't the final word.
Absolutely not. It's crucial to be aware of the limitations. For instance, they mostly used a naturally occurring mix of Tritrichomonas species found in the mice.
Which is realistic, maybe, but makes it hard to know which specific one is doing what.
Exactly. It reflects a real-world scenario, but it complicates pinpointing the exact role of each individual Tritrichomonas species in triggering this whole cascade. Future work needs to figure out which species are the main drivers.
Okay, that makes sense. What else?
Well, while they clearly showed that sphingolipids from Bacteroides were sufficient to cause the NLR family pyrin domain containing 6 response,
Wow.
the precise molecular details of how NLR family pyrin domain containing 6 actually senses these lipids, that's still a bit of a black box. Needs more investigation.
Right. How does the sensor physically detect those specific fats? We don't know yet.
Not in detail, no. And then there's that observation about female mice showing a stronger effect.
Yeah, the sex difference.
It strongly suggests there might be sex-specific factors, maybe hormones or other differences influencing this pathway. But again, that requires a lot more research to understand why.
Definitely. And the big one, I suppose.
Yeah.
This was all in mice, wasn't it?
Primarily, yes. And mouse models are incredibly valuable tools. They give us fantastic insights.
But we can't just assume it works exactly the same way in humans.
Correct. We always need that caution. These findings absolutely need further validation in human studies to confirm how directly relevant this specific mechanism is for human health and disease.
Okay, so bearing those limitations in mind, what's the sort of expert take on this? How is this research being viewed in the scientific community?
Well, I think the general feeling is that this work really challenges some long-held assumptions. The traditional view often focused on the immune system directly recognizing specific patterns on pathogens.
Like seeing the bug itself.
Right. And this study introduces this really fascinating alternative or perhaps complementary idea of indirect sensing detecting invaders by the changes they cause in the surrounding microbial community.
So the expert view highlights the microbiota, the gut bacteria, not just as bystanders, but as active participants.
Very much so, as intermediaries, really, signaling the presence of these protozoa through their metabolic byproducts like those sphingolipids.
And what about those goblet cells you mentioned earlier?
Experts are definitely highlighting their importance, too. These sentinel goblet cells, the ones rich in NLR family pyrin domain containing 6, and their ability to rapidly adjust the mucus barrier in response to these signals. That seems to be a really key feature for maintaining a healthy gut environment.
A rapid adaptation mechanism.
Exactly. Overall, the study just underscores the sheer complexity of the interactions between us, the host, and our resident microbes. These indirect sensing pathways might be playing a really critical role in maintaining that delicate balance and preventing disease.
And it probably opens up more questions than it answers, in a way.
Oh, absolutely. Good research always does. Experts are pointing out that this raises truly intriguing questions. If our bodies can sense protozoa indirectly like this, what other microbial interactions might be sensed through similar roundabout ways that we just haven't discovered yet?
Yeah, are there other examples of this kind of eavesdropping going on?
Potentially.
Yeah.
And what are the broader implications for immunity? Not just in the gut, maybe elsewhere in the body too. There's a lot to explore now.
So this deep dive, it really uncovers this fascinating hidden layer of complexity in our gut health, doesn't it?
It really does. When you think about that vast community of microbes inside you, it's crucial to remember they're not just sitting there.
Yeah.
They're constantly interacting, communicating with each other and with our own body in these incredibly subtle and sophisticated ways.
So the next time you're thinking about gut health, maybe considering your diet or, how you're feeling, it's worth remembering this pathway we've talked about. That bacteria can essentially act like an early warning system, alerting your body to other microbes, even ones your immune system might not see directly.
It's a powerful idea. And it really makes you wonder, doesn't it, what other subtle shifts in the gut environment, maybe triggered by diet, stress, medication, could be setting off important immune responses that we're not even aware of yet?
Yeah, how much does what we eat influence these bacterial signals, for example?
That's a huge area for research, how diet shapes not just the bacteria present, but also the metabolites, the signals they produce.
And this research definitely opens up doors for more exploration, maybe for listeners curious to learn more.
For sure. You could look into the specific roles of different Bacteroid species, perhaps, or dive deeper into the chemistry of those sphingolipids.
Or even just learn more about goblet cells and that crucial mucus layer and how they contribute to this whole system.
Absolutely. Ultimately, I think the takeaway is this surprising interconnectedness. The microbial world inside us is constantly talking.
And listening to that conversation, or at least understanding that it's happening, seems key.
It really reinforces the idea that maintaining a balanced, diverse gut microbiome is likely even more fundamental to our overall well-being than we perhaps fully appreciated before, especially when you consider these complex, indirect ways our body keeps tabs on everything.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.